identified as a stain from fluid from decomposition of the human body. Did your
Objection misstates the facts, Judge.
Does your test refute that?
Well, I can't speak to whether or not there was a blood or decomposition fluid present there because my test is negative, so I can't.
All we can say is your test did not detect whole blood cells.
It didn't react with any type of substance that was present there. Whole blood cells that maybe were, as we talked about, partially broken down. So I have nothing that would indicate that there was blood contained on those items.
But you indicated it could be decomposed fluid, including decomposed blood, that might not react.
Well, the test...
This is the testimony of the witness.
The
witness never testified to such a thing.
Objection sustained. Rephrase your question.
I guess what I'm trying to figure out is, does your test indicate that the stain was not the fluid from a decomposing human body, in your opinion?
Based upon my testing results, it's negative, so I can't testify to anything other than the evidence that was there that I tested was negative for the presence of blood, which I can't state anything other than I didn't detect any biological material, blood, or other fluid based upon my results.
Could the stain still be decomposition fluid?
As far as if there were other biological fluids that could have been present in there, it's possible.
Okay.
Based on my test, I can't say anything other than it was negative.
All you can say is it wasn't blood.
Correct.
Okay, I've got that straightened out. Now, what happens to DNA when cells decompose?
Well, as cells decompose, the DNA is going to start to also be degraded. Because if you think about where the DNA is housed inside the nucleus of a cell, if the cells start to break down, eventually it's going to get to that center portion, the nucleus, which is also going to cause the DNA over time to also start to be diminished, losing the information that's present.
And in order for DNA testing to work, you have to have intact strands of DNA.
Right. With the 13 locations that I examine as a part of my analysis, if I have portions of those locations still present on the chromosome, I may be able to obtain a result.
But generally with decomposing cells, you don't generally expect to find DNA.
Sustained.
I'm sorry, Your Honor, I don't believe she answered that exact question. It is cross-examination, Judge, so I would ask for some leeway to get...
One more time.
If I could forward to read back the question.
But generally with decomposing cells, you don't generally expect to find DNA?
Right, as the body breaks down, that information that's housed in the nucleus of the cell is also going to start to break down. And so the likelihood of obtaining a DNA result does start to diminish as the body starts to decompose.
Now, I want to go to this chart. I don't think I need to step down if you remember when it was written. I want you to clarify for the jury what this result means.
Yes, Mike.
This result that counsel wrote on this chart is 50, like 55 is RFQ. What does that really mean?
Okay, so the two numbers that are circled there, the 50 is what's referred to as the peak amplitude threshold, or the first level of detection for what's considered a peak in the FBI laboratory's DNA analysis unit, which is based on validation and testing to support what's a reliable result in the laboratory. So during the capillary electrophoresis process, when the DNA is injected into the capillary column and the DNA strands are traveling through that capillary column, at the end there is a laser. And those DNA strands have been tagged with a fluorescent tag, which enables then that laser to detect the signal of that DNA strand as it passes by. And that's the fluorescence that's then collected, and that's the end result, that DNA profile, based on how much fluorescent was detected at each of those locations, which is then generated into the peaks that we talked about at each of those locations. So at 50 RFUs, that's when the computer interprets if there's enough signal there to determine that a peak can be called. That's the first